one posted. So it records to the cloud and it takes a little while. And apparently I haven't changed my settings so that I can record it locally, which is faster. But that's okay. We'll figure it out. Okay, so we're gonna screen share to the PowerPoint. So you can, if you have questions, you can put it in the chat, or you can unmute yourself and say something, however you want to do it as we go through this. And I've gotten a little better at this, but it's still weird sometimes, so apologies for accidentally changing slides or something like that. Okay, so this is where we left off last time. We were talking about the different ways that muscles can contract and kind of the implications of that. So we have two basic contraction types: isotonic, where the tone of the muscle stays the same as the length changes, and then isometric, where the length of the muscle changes, or sorry, where the length of the muscle stays the same as the tension of the muscle changes. So we talked about this a bit already. So there's two kinds of isotonic contractions, because you can maintain the same tone while the muscle lengthens or while the muscle shortens. And I know that it sounds weird for it to lengthen, but we talked about how when we use a muscle as an antagonist, then we can be contracting it while letting it lengthen to better control the action that the agonist or the prime mover is doing, right? So this is kind of the standard one, the one that we think about when we think about how muscles contract, right? So the muscle contracts, it shortens its length while maintaining the same tension, and typically that's going to result in something like a flexion or an abduction or something. So that's pretty standard. This is the weird one, eccentric isotonic contractions. So here the muscle is generating tension. We have, sorry, lost words. It happens, but anyway, we're generating force, but we're actually letting the muscle lengthen. And so here we have a movement where this would be what's happening in the biceps as it lets the arm lower this weight, because we're controlling that movement instead of just letting your arm flop down and damaging your elbow or something like that. So we're still going through the contraction cycle, even as we're allowing that muscle to lengthen, which means that we're actually sliding our sarcomeres farther apart, even as we're letting them cross bridge. So very weird situation, but it's definitely something we do. It does have the highest risk of muscle damage because of the way we're, pulling them apart while we're cross bridging. And then we've got the isometrics, where we're just contracting the muscle without actually changing length. So this is the kind of thing that, like our postural muscles might do a lot of the time. And it's also like when you look up things like exercises you can do while sitting at a desk, you're going to find a lot of isometrics because you literally don't have to move while you're still working your muscles. So that's it's kind of a weird way to use it, because it's not like the real way, like when you're just doing things in your life, you're not usually like sitting there and like flexing your abs for no reason. But it's basically using your muscles in a way that we usually do when we're maintaining posture or things like that, or resisting something that's too heavy to actually move, that too. All right. Okay, so this kind of ties together the stuff that we talked about last time. So the total force of a muscle contraction ultimately depends on the total number of cross bridges and that's going to typically be in cross section. So the total number of cross bridges that we are bringing to bear dictates how much force a contraction has. The way that we get more cross bridges to form are a couple different ways. So the number of muscle fibers recruited, obviously the more muscle fibers we recruit, the more cross bridges we can form, right? And we call that spatial summation or multiple motor unit recruitment. You remember that one, MMU. Another thing that will get us more cross bridges is the size of the muscle fibers. So a muscle fiber of larger diameter, so like this is a small muscle fiber diameter and this is a large one, literally going to have more myofibrils in it, and therefore there's going to be more cross bridges being able to be formed. Okay? And pretty sure we already talked about this. Remember that the recruitment of different muscle fibers, we always start with the smaller ones, and then we recruit the larger ones, and the last ones to be recruited are going to be the largest diameter. So the more muscle fibers we recruit, the more force of contraction for both of these reasons. Other ways to get more cross bridges formed, increasing the frequency of the stimulation. So remember temporal summation, which can get us to muscle tetany. So tetanus, because we're not letting it relax, and that's as much force as we can generate from that number of fibers. The last thing that determines the force of contraction is actually how stretched out the muscle is when it starts to contract. So degree of muscle stretch is the last factor. And that's going to bring us to our next concept, which is called the length tension relationship. So this is a nice little diagram summarizing the last slide. This is literally everything. And we're going to talk about this part more, because we've already talked about everything else. So we just have to tackle this length at initial contraction. So the length tension relationship. So basically we know that as long as the contraction cycle is going, we're going to generate tension in the muscle fiber. But how much tension can occur depends not only on how long the contraction cycle is running, right? Because we know if it runs longer, we're going to keep generating tension, but it also depends on how long the sarcomeres are to start. And that makes sense because we know this is a sliding filament method. And so if we're starting relatively stretched out, and then we have a lot of sarcomere overlap that can still occur to keep generating tension. But if we start already pretty overlapped, there's only so much more sarcomere that we can shrink down. And so that's going to, restrict how much tension we can generate. So we know that we need our myosin heads and our actins to be overlapping. So we need our thick and thin filaments to overlap to have contraction. If sarcomeres are too stretched out, then not enough overlap occurs, and we can't generate enough tension. So you can think about, like if you're trying to grasp somebody's hand and you just have their fingertips, you're not going to be able to really get a lot, versus if you have a decent amount of overlap between your fingers and theirs. So when you're starting too stretched out, there's only a couple of cross bridges forming, and we can't really generate a lot of tension. It takes a while to slowly creep back together to get enough overlap. But when we're too contracted already, then there's just only so much more overlap we can generate. So this is also going to decrease the amount of tension that we can generate. So too stretched apart is bad, too close together is bad. And that means that where we want to be is somewhere in the middle, okay? It's very much a Goldilocks situation. So we call this the optimum resting length, which is where literally the length at which, when a contraction cycle starts, the muscle fiber can generate the maximum amount of tension. And the interesting thing about muscle tone, which we talked about before, right? That constant small amount of contraction, the nervous system is in charge of it. It typically keeps our sarcomeres at the optimum resting length. So when we say that, you know, one of the functions of muscle tone is to allow muscles to be ready to go when they're needed again, it's because they're keeping them at optimum resting length, and so we're able to generate maximum tension as soon as they're stimulated. So we of course have a little diagram for that, because what is physiology without graphs? So this is showing you the length tension relationship. Now you do not need to know these numbers, okay? But I'm going to talk about them just because it lets us communicate about it. So this is tension in percentage. This is length in percentage. So we have 100% over here, and we have over 200%. See, this happens sometimes, sorry. And also apologize about the dog. There's no stopping it, but the door is closed. And that's the best I can do. So you can see that if we stretch a sarcomere out, we can't get to 200%. We can't double the length of a sarcomere. We lose all overlap and we can't form cross bridges, okay? So we have to get below about 150% overlap, and you can see they're showing you the overlap down here, before we can have any, or length of the sarcomere, before there's any overlap at all. So at this point, we can actually generate cross bridges, but not very many. So this is still too stretched out. So we can see that optimum resting length is, again, you don't need to know the numbers, is pretty much between 90 and 120% of sarcomere length. If you get too stretched out beyond that, then you can't generate maximum tension when you initiate from there, like it'll take too much time. And if you overlap too much more than that, then there's just not enough overlap to generate maximum tension, okay? So we can see though that the too much overlap cuts our percentage of tension a lot faster than the stretch out. So you're definitely better off a little bit more stretched out than you are too much overlap. Okay, so this is the optimum resting length. And what you want to remember here is that it's in the middle. Okay, very much a Goldilocks principle. We don't want to be too short. We don't want to be too stretched out. We want to be just in the middle, just right, in order to generate maximum tension. Okay, so something we're going to talk about more this week is the trade-off between power and speed. We're going to talk about levers on Wednesday, and that's very much going to be about speed versus power. Because if you're trying to maximize one, you lose the other, and vice versa. So just like we have this force-tension relationship, we also have a force-velocity relationship. So we know that we get more force with more cross bridges, right? We've already talked about that over and over again. That's what we just discussed. But cross bridge formation actually takes time, and it will slow the basically the speed at which the sarcomere overlap will occur. So basically we're slowing down the sliding process if we're maximizing the number of cross bridges. So this is where speed and power are a trade-off. So if we want a muscle to contract fast, we actually aren't going to be able to achieve maximum force because maximum force takes too long. So if we want to increase the speed or the velocity of sarcomere overlap, we actually need less cross bridge formation, and that means we're actually going to end up with less force. And which one we want depends on the situation, as we'll see when we get to the lever systems on Wednesday. I believe I have a graph. It's not nearly as fun, but it's what I could find. So this is showing us the trade-off where this is speed or velocity, this is power, and this is force. So force is what we're generating with cross-bridge formation. And what we're getting here is maximum power. And as we increase the velocity or the speed of contraction, we can see that we're losing force and losing power. So all I want you to take from this is that, again, there's a trade-off between speed and power. Okay, and it's because of the time it takes for cross bridges to form. And we're going to look at the physics of the lever systems and how that also gives us a trade-off between power and speed. Okay, so the last thing we want to look at here, I think it is, we'll find out, is the idea that there's actually different types of muscle fibers. So muscle fibers, even though we've been talking about them all, like, oh, we have skeletal muscle fibers, they're not all the same. They're actually different types, and they're different types for different reasons. So we categorize them or classify them by two things: the speed of contraction and what source they use to make their ATP, so their preferred metabolic pathway. So when we do that, we actually end up with two classes and three total types of muscle fibers. So we divide them up by their speed. So basically how long it takes them to reach peak tension when stimulated. So remember, a muscle twitch is that single peak of tension in response to a single stimulation. And you may or may not remember, but I showed you a graph of different muscles in the body. Like there was an eyelid muscle and there was a calf muscle, and like the eyelid muscle was super fast and it twitched really fast. And then like the muscle in your lower back was like, it took forever to get to peak tension. So this is why that happens. So slow twitch fibers, the ones that take longer to reach peak tension, are called slow twitch or slow fibers, and we also call them type 1 muscle fibers. So of course, this is another situation where we have a bunch of names for the same thing, because they've been studied extensively and different people called them different things. To contrast that, we have fast twitch fibers, and they are also called type 2. Now there's actually one type of slow twitch, and there's two types of fast twitch. So we're going to look at all three of them and compare and contrast them. So slow twitch fibers, of course, have additional names. So they're also called slow oxidative or red fibers. Now, slow oxidative is probably the most useful name because slow twitch fibers are known for always using the aerobic metabolic pathway to generate their ATP. So they're always going to be very efficient in their ATP use. Because they're using the aerobic pathway, they need to have a guaranteed supply of oxygen. So these guys typically have really good blood supplies, so lots of capillaries. They have a lot of myoglobin, which allows them to store oxygen for when their blood supplies get cut off. This makes the fibers red, which is why they get called red fibers. They also have lots of mitochondria because, of course, the only way to have aerobic respiration is by having mitochondria. Now, because they're so efficient at generating ATP, they tend to be pretty fatigue resistant. And so we see them a lot in postural muscles. So muscles of your back, muscles in your legs that are, that help you stand, anything, muscles in your neck that help hold your head upright, anything involved in maintaining yourself in an upright position is going to have a lot of these slow oxidative fibers. Now, Oh yeah, and one more thing. So they're typically thin. They have a very thin diameter. So these guys are our smallest diameter fibers. And that's because we want to easily diffuse oxygen through the whole fiber. So we need it to not be very deep because we want oxygen to get to all the mitochondria. So the downside, it's not really a downside, but the one thing about them is that it can take up to 100 milliseconds for them to reach peak tension, which is pretty slow for skeletal muscle. So this is where we get the slow part of their name. And then oxidative comes from the fact that they're always using oxygen. So they're efficient. It takes them a while to build up to peak tension, but they can also last for a really long time. So these are our endurance muscle fibers. So now we're going to look at how that compares to our fast twitch muscles. So the common kind that we have two types, but this is the common one, is also called fast glycolytic. So fast referring to the fact that they reach peak tension really fast, within a few milliseconds. And glycolytic because their major source of ATP is all of those anaerobic pathways we talked about before. So they typically burn up all the glycogen stores. They will try to keep as much creatine phosphate around as possible. They're not going to worry about having mitochondria, a good blood supply, or a lot of myoglobin because they're not relying on oxidative sources of metabolism. That also means that they get to be fairly thick, and their appearance is actually typically white because without the myoglobin to hold oxygen, they look pretty pale. The other thing is that glycogen is also a fairly pale molecule. So between the glycogen stores and the lack of myoglobin, these are white fibers. Now the downside to anaerobic sources is, of course, it's inefficient. You use it up pretty fast. And so they don't last very long. So rapidly fatiguing type of muscle fiber. So when you want things to go really fast, but you're going to go with these guys. They also have the most extensive sarcoplasmic reticulum. They're fast at releasing their calcium, so this helps them be faster twitch. But it's not going to last that long because there's a downside to rapid generation of ATP. It's that you run out really fast. So these guys are the opposite of slow oxidative. Now there is a second type of fast twitch muscle fiber. It's fast oxidative glycolytic, which I think it's funny, but we abbreviate that to FOG. So these guys have a little bit of both. They're trying to be the best of both worlds. So they're really good at reaching peak tension fast, but they're better at using an oxidative pathway than the fast glycolytics. So they're a little bit, they're faster than the slow oxidative. They're better, they're longer lasting than, they're longer lasting than the fast glycolytic. So they're kind of in between. We don't actually have a lot of them. They're more common in other species though. So clearly it's not super critical for us to have them. But it is the third option. Let's go have words with that dog. Okay. Now, even though we have, postural muscles that need to be fatigue resistant and that need to be really good at contracting for long periods of time, and other muscles that need to be really fast, most muscles are actually mixed. They're a combination of the different fiber types. Now, depending on a muscle's job, they will probably have a larger proportion of one fiber type versus another. So like eyelid muscles, facial muscles, things that just they're meant to not be going all the time. They're going to have more of those fast glycolytics. I'm sorry, there's gardeners outside. Give me 1 second, and then I will complete this thought. Must pause. Okay, that'll be a fun little blip in the recording. Okay, so so muscles that are meant to just be really fast and then relax again, they're going to have a lot more fast glycolytics. And then postural muscles, you know, walking muscles, muscles that have to do work for long periods of time, they're going to have more slow oxidative. The exact proportions vary by a lot of different factors though. So we know that there are genetic differences between fiber distribution types. We know that there are sex differences. There's other factors. There's some amount of influence you can have with training. So people that train for marathons are going to have more slow oxidative. People that train for, you know, more sprinting related activities are going to have more fast glycolytic. But one thing is always true. Motor units are always a single fiber type. And we can tie this back to what we learned about with multiple motor unit recruitment. So here's a picture of muscle fibers. So all the darker ones are slow oxidative, and the lighter ones are fast glycolytic. And maybe there's some fast oxidative glycolytic in there, I don't know. But for the most part. So again, the presence of myoglobin and oxygen makes these guys darker, and the lack of myoglobin and the presence of glycogen make these guys lighter. The diameter differences might not appear that profound to us, but they definitely make a big difference. Of course, she's back upstairs. Okay, so this is a nice summary table of the differences between them. So you can definitely make your own for comparing and contrasting the three different types. As far as the differences, you can see this covers pretty much everything we talked about as far as what we're using to make ATP. Are we using oxygen or not? How fast are we contracting? How fatigue resistant are we? Myoglobin and glycogen and color then go back to what we're using for ATP. So glycogen corresponds to anaerobic, myoglobin corresponds to aerobic, and color is going to correspond to myoglobin. I would use other colors, but it takes too long to switch between them, so it just ends up being a bloody mess basically on the slides. Okay. And then I found this cute little chart too. So this is a simpler summary chart, but it kind of shows you the same thing, right? So our slow oxidatives are going to be more in play here. Our fast oxidative glycolytics in the intermediate, and the fast glycolytics for the fast stuff. I know, okay. So before I get to the next thing, let me just go back and say one more thing. So we talked about the multiple motor unit recruitment before, right? And we know that motor unit recruitment operates by the size principle, where the smaller diameter fibers are recruited first. So, given what we now know about the different types of fibers, we know that slow oxidative motor units are the first ones recruited whenever muscles are activated. Even though they're slower to respond, we know that they're going to last for the duration of whatever muscle activity we need. Now, as we're trying to generate maximum force, we have to start recruiting fast twitch fibers. And so when we're trying to generate maximum muscle force, that's when we bring our fast glycolytics to bear. And the reason why we don't want them until the very end, until we're generating peak force, is because they don't last that long. And so unless we're generating maximum tension, which isn't going to have a great duration, we don't want to bring them in because they just don't last very long. Okay, so trade-offs, everything is trade-offs. So in this case, we're trading off between fatigue resistance and speed of twitch. All right, So the last thing we want to cover for this chapter is how using muscle or not using muscle affects muscle mass. So we're going to talk about terminology, so hypertrophy, atrophy, and how we can build muscle. Did not actually put anything about performance enhancing substances in there, but it's in the book if you're curious. So resistance exercise, so we talked about the different types of exercise before, right? Because they have an effect on skeletal structure. So resistance exercise, which is the best at building bone density, is also how you increase muscle strength. And it's for the same principle. You're telling your muscle to try to work with more than it's really good at. And that means it has to get stronger in order to respond to what you're asking it to do. So when we start asking more of our system, they know to respond by getting better at being used. Now, we don't see a lot of mitosis in skeletal muscles. Our skeletal muscle fibers are not capable of it. We have a small amount of stem cells that can respond, but mostly they only come into play when muscle is actually damaged. So when we build muscle bulk, we're actually doing it by a process of hypertrophy. So muscle fibers will thicken by making more myofibrils. They will actually grow the myofibrils, they'll split in half, and then they'll increase their diameter, and will thicken the entire diameter of each muscle fiber. So we don't necessarily build by putting more muscle fibers in, we just increase the diameter of the muscle fibers, and that still increases the total cross-sectional area. And that means we have more more cross bridges and therefore more total force. Okay, so hypertrophy is increasing the size of something without increasing the number of cells. And this is the textbook's picture for resistance exercise. This guy has really worked on his biceps. Now, we can contrast that with endurance exercise. So we call this aerobics. And so when we do more aerobic exercise, we're increasing our resistance to fatigue. So this is not how you bulk up your muscles. You're not hypertrophying anything. What you're doing is encouraging your muscles to enhance oxygen delivery. Basically, you're telling your muscles, we need more fatigue resistance, so we need to support our slow oxidative fibers better. And so our muscles can actually increase the blood supply. They can grow more capillaries around the fibers. They can actually increase the number of mitochondria in the fibers and increase the amount of myoglobin in the fibers so that we're storing oxygen, we're doing more aerobic respiration, and we're delivering more oxygen and fuel to these fibers. Okay, so this is a faster process too, so it's easier for us or quicker for us to increase our aerobic capacity than it is to increase our strength. It takes a little bit longer to do that. And we're not covering cardiovascular in this semester, but this is also applicable to heart muscle. So we can increase our heart muscle efficiency by doing this as well. So just like resistance exercise is also good for your skeletal system, as well as your muscular system, endurance exercise is good for your cardiovascular, as well as your muscular system. And that was that there. And I didn't put an actual slide in for atrophy, but atrophy is the opposite of hypertrophy. So atrophy means decreasing the number of myofibrils, which results in decreasing muscle diameter. So this can occur because of disuse. The less we use our muscles, the less muscle we're going to have. It's just like the less we use our bones, the less bone we're going to have. Our body is efficient, and if it's not going to waste time maintaining big muscles if we're not using big muscles. But this can also occur when there's nerve damage. So if you lose the connection between your nervous system and your muscles, you lose that muscle tone, and you lose the constant signal that's telling your muscles that they're still functioning. And so you can get a very rapid atrophy of muscles when the nerves are no longer communicating with them. And we see that with people that have like spinal cord injuries, especially, like they have to do a lot of physical therapy to maintain muscle mass in their legs. If, you know, if it's like an injury where they lose use of their legs, because there's no longer any nerve communication telling their leg muscles that they're being used still. All right, does anybody have any questions about this chapter 13 stuff?
Is this chapter 13 or chapter 10?
I have no idea what chapter it is, sorry. Lecture 13.
Oh, okay, lecture 13, okay.
No, you don't have to know anything about the performance enhancing substances.
Oh.
If you're curious about it, you can read the little bit that's in the chapter. I don't feel like it's relevant enough, and I usually don't have time to cover it anyway, so I skip it. We could do like a whole lecture on that, you know, but you guys don't need to know it for our class. Any other questions? All right. Okay, so I'll stop us here. We don't have enough time to start the next one. So we will cover the next lecture, which should cover, it covers the next chapter, but some of it's not really in the chapter. So I kind of had to pull it from other places. But basically, we're going to talk about some of the details about smooth muscle that we didn't address before. So we're going to compare it to skeletal muscle now that we know about how that works. And then we're going to talk about lever systems. So, so yeah, we'll do that one on Wednesday. In the meantime, I'm gonna stop the recording. I'm not done yet though. I'm gonna cover the lab stuff, but just, you know, let's take a 10-minute break. Well, it's 9:38. Take a 10 to 20-minute break, and then we'll do that at 10. Okay, so if you want to stick around and get the lab information live, we can do that. If you need a break from staring at screens or whatever, then do that. I'll be posting everything on YouTube. Okay? I'm gonna take my dog outside for a couple minutes and hopefully she stops barking. Okay, so I'm not gonna turn any of this off. I'm just gonna mute and turn my camera off and stuff, and then I'll be back before 10. Okay? All right, let's turn the recording off. Do you want to stop cloud recording?